Hello, I am Eun-Hye Lee, the first author of this paper from Gyeongbuk National University, College of Medicine. I'm delighted to introduce this paper through this video interview.
I'm Yun-Sok, the co-first author of this paper.
I'm Tae-Hwan, the corresponding author of this paper.
Let us briefly introduce the content of this paper.
This study aims to analyze PDK4 expression in prostate cancer cells and human samples and to explore the gene's clinical significance.
Prostate cancer ranks as the second most common cancer in the men globally, representing a significant cause of cancer-related mortality. Prostate cancer is the fourth most common cancer in Korean men. For those aged 65 years and above, lung and prostate cancer were the most common in men in Korea. The incidence and mortality rates of prostate cancer tend to gradually increase over time.
Major reason of difficulty in controlling cancers is that cancer cells gain evading ability leading to metastasis. Cancer metastasis is the process of dissemination of cancer cells from primary site to others. In early stage of prostate cancer, it develops slowly and locally remains. In late stage, cancer cells cross the cancer capsule's barrier and invade to other organs such as bones, liver, lungs, and lymph nodes. The most common location of metastasis and metastasis requires complicated treatment and a decline in a patient's quality of life.
Pyruvate dehydrogenase kinase 4, PDK4 is a member of pyruvate dehydrogenase kinase, PDK, that postulates pyruvate dehydrogenase, PDH. PDK controls the switch of aerobic glycolysis by regulating PDH. PDH converts pyruvate to acetyl-CoA to generate cellular energy for cell proliferation. Control of pyruvate oxidation is primarily attributed to PDK-dependent PDH phosphorylation. PDK4 prevents pyruvate from entering the 3-carboxylic acid cycle, so inducing aerobic glycolysis to facilitate the survival and progression of cancer cells.
Previous research of PDK4. In lung cancer cells, the inhibition of PDK4 reduces cell migration and invasion. In colorectal cancer cells, the inhibition of PDK4 reduces cell migration and invasion while increasing apoptosis. PDK4 shows significantly higher expression in breast cancer tissues. In breast cancer cell lines, microRNA 211 was found to suppress PDK4 expression, leading to the induction of apoptosis. PDK4 was found to be overexpressed in ovarian cancers. It is associated with chemotherapy resistance and poor prognosis. It was confirmed that PDK4 promotes cell proliferation, migration, and chemotherapy resistance in ovarian cancer cells.
We conducted PDK4 expression experiment on blood cancer. MS-based quantitative proteomic profiling of phosphorylation was analyzed using a Venn diagram and a volcano plot. The results showed that ANXA2 and MPGL1 were detected as substrate, FIN kinase were detected, and both SRC regulatory kinase and SRC were detected.
We proceeded to confirmation of suppressed PDK4 expression by using siRNA treatment on DU145 and NK cells to clarify the role of PDK4 in prostate cancer. PDK4 knockout in NK cells suppress their invasion ability. Thus, PDK4 inhibition may play a role in hormone-sensitive cancer. PDK4 knockdown increased E-cadherin and decreased vimentin levels in Western blot analysis. PDK4 suppression downregulated phosphorylated SRC, the activated form of SRC.
This established the baseline characteristics of the 77 patients enrolled in the study. We examined the expression of PDK4 in BPH, hormone-naive radical prostatectomy, neoadjuvant hormone therapy, radical prostatectomy, and CRPC. A tumor progressed from BPH to CRPC, and the PSA level increased. The PDK4 MR expression level also increased. However, PDK4 was not related to tumor staging, Gleason score, and biochemical recurrence.
For PDK4 expression, we proceeded with qPCR and IHC staining using patient samples. As we expected, PDK4 was highly expressed in prostate cancer that was becoming aggressive, and as PSA level increased. PDK4 knockout in NK cells suppressed their invasion ability. PDK4 was related to the progression to an aggressive stage, such as metastasis, rather than cancer proliferation. PDK4 was highly expressed in prostate cancer that was becoming aggressive, and as PSA level increased.
PDK4 is thought to play a significant role in hormone-sensitive prostate cancer, and concurrent targeting of PDK4 is anticipated to further enhance the therapeutic outcome. This finding not only enhanced the understanding of prostate cancer pathogenesis, but also of a potential view for developing target therapy. The relation between CRPC and PDK4 needs to be explored further in the future.
In summary, this study demonstrated that PDK4 is overexpressed in prostate cancer tissues and potentially involved in the regulation of cancer cell invasion and the progression to CRPC. Further research into PDK4-mediated mechanisms in prostate cancer progression hold promise for the development of novel therapeutic interventions aimed at improving patient outcomes.
Thank you very much.